before we get into the exciting realm of genome nomenclature. Okay, so we are actually writing a grant. This is the provisional titles: AgBioData, a coordinated, cooperative, and innovative network. Wait a second, my screen is obscured here. Of genomic, genetic, and breeding databases for enhanced agricultural research outcomes. This is funded by NIFA under USDA. The initiative is the FACT and Agriculture Cyber Informatics Tools and Initiative. And I've included a link there for you to get more information about it. That tells you what FACT is. And under FACT, we are doing a coordinated innovation network, which is which projects should foster communities that address critical areas by bringing together experts from different disciplines to identify solutions. So that's what we are doing. Now, we submitted a letter of intent July 25th, and we are sorry we didn't get approval from the rest of the group. We were very rushed about getting that letter in on time. One thing the USDA said is that they cannot accept late things, even if they're 1 minute late. So they're very picky about their rules. So we are very lucky. Our letter of intent was accepted. And Dory Main is going to be the project director. She's actually the only, at Washington State University, she has people to help her, distribute those funds and everything. And if funded, the funds will go to Washington State for a yearly workshop and website support, which will fund 10% of a person. Iowa State University for a half-time coordinator for the whole AgBioData project, which will be Jackie Campbell. And then some money will go to Phoenix Bioinformatics for a database sustainability pilot study. I'm going to tell you a little bit more about that. That's why Ava Hawala is on the grant. So the objectives, I did send out the letter of intent to everybody, but if you haven't, these are our objectives. We want to develop and implement standards for AgBioData data curation. And we'll decide what that means if you're in that working group. We want to establish common practices for a broad use of ontology and provide tools and training for researchers. So this doesn't mean we're going to develop any ontologies. This just means we want to sort of evangelize for their use of ontology in the database community and the larger research community. We want to establish Metabase standards across AgBioData member databases and promote compliance. This is the part where Phoenix Bioinformatics and Ava Hawala will help. We want to identify opportunities. Oh, no, I'm sorry, wrong one. Forgive me. We're going to identify opportunities for a federated model of data exchange for AgBioData members. This is the one Phoenix and Ava Hawala will be involved in, is identifying funding options for long-term database sustainability. And what Phoenix is going to do is a pilot study, some basic, informational research on how could we get money from, if everybody that writes a grant writes in some money for the appropriate database, could that money be funded to an outside resource like Phoenix Bioinformatics and then given back to the databases somehow without having to go through a sponsored project office at a university and where they would take 50% or whatever. It's that kind of thing. I think this is a real good objective. And then lastly, working with funding agencies and journals to enhance data provision by researchers. So those are the objectives. The timeline for writing this grant is very fast. The steering committee is going to get a draft to the whole group by September 15th. We have until October 19th to submit. It's got to be done by October 19th. It's due at the agency October 30th, something like that. So we are really hoping that we will get a lot of interest and people can help us, sort of based on, those are really the old working groups. Most of those correspond to a working group. So we would love to have help between September 15th and the 19th. And if you're so excited and you can't wait and you would like to be involved with the first draft, just let me know, and we will certainly include you. Now, also, this is an exciting thing. So we can add official collaborators and named personnel. We would like to add a representative from each database as an official collaborator on the proposal. And so we really want to get that one contact person from each database. And I'll be sending letters to each of the people that I think it is, but we would like to get that list of everybody soon. So, whoops, now I can't go to the next slide here. Oh, there we go. Okay, and a note about the steering committee, because remember at PAG, we asked for volunteers to come onto the steering committee. And we've been working pretty hard, first on the white paper and now on this. So we're slowly thinking about and writing governance bylaws for the AgBioData group. And so we will rotate people onto the steering committee. It might be by election, and we hope to start this as soon as the grant is in. So I do apologize this is taking so long, but for those of you that have volunteered, I know who you are, and I'm excited that you're willing to join. So we just want to get this sort of formalized. Okay, now, are there any questions about the grant and the grant process? I know everybody's muted, but if you do have questions you do want to ask.
Lisa?
Yes, Jack.
What agency? I must have missed that. You're going to?
It's NIFA under the USDA.
Okay, thank you.
Mm-hmm. So for today's meeting, which we're going to try to finish in 1 hour, we want to talk about genome nomenclature. And I did send out emails about what I mean by genome nomenclature. And I'm hoping that we might be able to come together as a group and establish some guidelines that we could use, at least AgBioData wide. This is an emerging issue, and it's very, very important and could be very confusing. So we're going to hear short presentations from each of these groups, and we're also going to be recording the session, which I think is okay with everyone. I didn't hear from Marcel and Pankaj. Pankaj, I'm sorry, I misspelled your name. I did get your email.
I did respond. I'm fine with the recording and sharing my slides.
Okay.
Same here. I did responded to your email.
Oh, you did? Okay. Okay. Perfecto. And Tanner may give us a few verbal comments about wheat. So without further ado, let's begin with maize. So I'm going to stop sharing my screen. And Effie and Maggie, can you take over there?
One quick question. Is anybody recording the meeting?
Yes.
Thanks.
Yeah, and it will be available on the website.
Okay, can you see the presentation? You probably see the wrong page.
I see it in presentation mode.
Yeah, let me see. Fix that real quick. Okay.
Perfect. Perfect.
All right, so we'll try to zip through this quickly. I'm going to do what we have done, and Maggie's going to talk about what we need to do next. Okay, so in Maize, we are at the moment, MaizeGDB has 10 complete reference quality genomes. 3 of these have multiple versions, and then we have 4 that are pretty close to done, and then we have 25 plus that are in progress. So we're just kind of getting deluged here. And we knew this was coming, so a few years ago we looked at this and said, well, we can't just leave people to name things on their own, or we're going to have a mess. And then the Maize community has this long history of setting nomenclature rules, and mostly of following them. There's a nomenclature committee for Maize. And then the committee in MaizeGDB attempts to enforce these rules, but these have been mostly for locus names and phenotypes and whatnot. So the genomes and gene models was new. So where we were when we started this process. We only had the B73 reference genomes, but we had 3 versions. And the version names were consistent, although they didn't give a whole lot of information about what was actually being the genome. The annotation names were 4A, 5B, 5B plus, and the annotation names didn't really give you any clue as to what genome they were attached to. And I have to put in a plea: do not use a reserved URL character in your identifiers. That the plus and the 5B plus still occasionally breaks code in the MaizeGDB website. But although it did work, but we could see that this was not going to work for much longer. So we laid out our objectives. The names had to obviously be unique. We wanted them to be consistent. We wanted both human and machines to be able to make sense of the names. We wanted them to be short. We wanted to be able to enforce them and to avoid reserved symbols like the plus. And so this was surprisingly difficult. This looks pretty straightforward, but it turned out to be quite a challenge to pull this off. So with the genome assemblies, we decided to go to more information-rich names. There's a fair bit of controversy, differences of opinions. Purists say that the identifiers should contain no information in them and should be completely random. And the biologists like to know what they're looking at. And so with the identifiers, we decided to go the information route. So they're built up like this: Z, because all of maize will be a Zia; the specific species, a single letter; the cultivar accession, whether it's a draft or a reference, the group that did the assembly, and its version. So current B73 reference genome is ZM maize. B73, it's a reference quality. The gramine is the assembly, and it's 4.0. So then we had the genome annotations, and we wanted to use the annotations as a prefix. So that meant they had to be short, because we didn't want to stick all of that previous stuff in front of a gene model name. But again, they needed to be unique. We wanted the identity of the genome assembly built into the name. But we wanted to keep the information pretty minimal to keep it short. So what we came up with here is, again, the ZM, the Z with the species, single letters species, the series, which is just sequential, and then the assembly version represented by a letter. So for maize line W22, this was the 4th genome that was sequenced since we started the conventions, and there are 2 versions. So the first one is ZM0004A, and the second one is ZM0004B. And then this becomes, and then the number representing the gene models is then attached onto the end of this identifier. For third-party annotations, we're going to accept whatever conventions they use. These are usually add-ons, additional information. We're not trying to enforce naming conventions outside of what you might call the official annotations.
I'm just going to say you're approaching 5 minutes here.
Okay. Gene models. I will skip this because I pretty much said that. So versioning, we do have multiple versions of the same, of an annotation, the same assembly. So we assume that the names are going to stay the same. Split or merged gene models must get new names, and versions need to be explicit. So we added dot for the name of the annotation, but this dot notation does not get carried over into the gene model names. Let's see. Okay, so now I'll give it over to Maggie.
Okay, so I'm going to talk about stuff that we would love to have a later discussion on with TAIR and other groups, because as they start having to deal with pan IDs. So a pan genome, of course, is the cumulative diversity within all sequence cultivars in the species, in our case, the maize, and that includes all annotated genes within each cultivar. A pan gene is a gene that has an ortholog in multiple maize cultivars within the pan genome. So one of the things is, how do we name these? We have several ideas. None of them are written in stone. These are just some of the things that we've talked about at MaizeGDB, and we hope to have a discussion with the nomenclature committee in the future. So for curated or human curated genes, and this includes genes that have been characterized genetically, pan genes of curated genes will be given the approved gene symbol, such as ligulus 1, in every maize genome where that gene is present. For non-curated, or genes that have been identified through an automated process, no human curation, these gene models across all maize lines that are in the expected syntenic region will either be given the gene model ID of the first genome sequenced that has the syntolog, or be given a new identifier such as Z0, 1, 2, 3, 4, 5, 6 that will be shared as an alias or synonym across all the syntologs. These syntologs will still retain their unique annotation identifier too. And so yeah, those are the things that we need to address moving forward about pan IDs. And that's it.
Okay, give it over to the next one. Who's next? I'm not sure who is next. I can't remember.
I think that's me.
Tanya, is that you? Yeah.
Yeah, that's me. Let me know when you see my screen.
I can see it, but you might put it in presentation mode.
All right.
Thank you.
Arabidopsis so far was sequenced in 2000, and the original annotation, or the original sequencing was spread out over several different members of the consortium. And the initial annotation was done by the Institute for Genome Research, and they did 5 releases in total, and these were adding more information onto the same original chromosome or back sequences, assembled sequence. So there is one genome sequence that has been improved over time. From TAIR, it went to TAIR, a different group that did that for 5 releases, and the last release was done by yet another group at JCBI, and that's the current release that's out there. We don't yet know when the next one is going to be. The sequence came from Columbia-0. For the original sequencing, multiple Columbia stocks were used depending on which group was sequencing which chromosome. And it's unfortunately unclear how these are related to each other. So the proposal is for any future sequencing that will be done, but the reference seed stock will be this particular stock from the stock center. So everybody knows which germ chromosome is used. The naming convention for loci and genes, many of you are familiar with this. The locus is a chromosomal position with a start and a stop. Something kind of from here to here. And that is named with AT, Arabidopsis thaliana, 1 through 5, or which chromosome it came from. C, if it's a chloroplast encoded, and M, if it's a mitochondria encoded. G for gene, and then a 5-digit number. The locus is named that way. The gene models, which represent the different splice variants, or the single spliced version, is named with 8.12 or however many there are for a particular locus. For Arabidopsis right now, we don't distinguish between a genome assembly, meaning all of the individual pieces that were lined up to make the pseudochromosomes, and the gene annotation. I think this is not an ideal approach because what happens is that, for example, for the TAIR-10 genome assembly, the chromosomes, those are exactly the same as the chromosome sequence that were in TAIR-9. But the genes that are called and annotated individually upon that genome assembly are different in TAIR 10 than from TAIR 9. So what we've learned is that it's beneficial, and you probably realize this as well, to be able to name and version the genome assembly with a different string than the gene annotation. Existing upcoming issues, briefly, Columbia is not the only ecotype out there. There are many, 1001. But the Landsberg sequence was published, I think it was last year, and they used a similar naming approach, meaning using the AT1G number scheme. However, the Columbia, for example, 040, is a different gene product than the Landsberg 040. And that can be confusing. So together with the authors of the sequence, we were able to create a mapping file and make that available on TAIR and anywhere else that wants to show the mapping file as well, so that people can go between one ecotype and the other. What's coming up, or what's already in the works, is that there are several sequencing efforts using PacBio technology to create platinum standard, really high quality sequence versions from many different ecotypes in Arabidopsis, including Columbia-0, where they're going to de novo assembly the genome. And they're going to need we're going to need to come up with a consistent nomenclature approach across those different ecotypes. At the same time, there are many ecotypes in Arabidopsis that have already been assembled, that have already been sequenced as part of the 1001 Genomes projects, and those have been assembled using Columbia-0 as a reference, using that to guide the assembly, and they could and should be annotated. Finally, we have issues similar to those that many of you will encounter, which have to do with variation in gene insertion, deletion, rearrangement, or just nucleotide changes when comparing the genome sequence of any of the other ecotypes to Columbia. So handling that naming is a challenge. I got one more slide, which is what I thought I'd bring up about handling user feedback. So slide, I said, there have been 11 releases, and those genome releases are pushed into NCBI, into RefSeq on in the past it was annual basis, but now it has been larger gaps in releases. And what happens is that in between the releases, we get feedback on, oh, this particular gene model needs to be changed. I found a new gene here. And handling that propagation of information can be challenging because we want to communicate to the rest of the community, here are the new changes, and yet updating in all the various places that the information flows out to needs to be carefully coordinated because it is a big amount of effort to integrate new information. So that's an issue that I wanted to raise that I know that you probably are encountering as well. And that's the end of the Arabidopsis story.
Wonderful. A lot of similar issues already.
Mm-hmm.
I think we'll next hear from Suk.
Okay.
At least it's nice with Zoom that it is possible to switch presentations like this. It's a little stressful, but it's functional.
Okay, how do I do that? Like
at the bottom of your screen, there's a green share button.
Share screen, okay.
Yeah, and you could just share the application, then it'll be a little bit larger.
Okay.
There we go.
Can you see it? Okay.
Perfect.
So I'm going to talk about genome naming guideline in GDRs. I'm mostly using the examples from GDR, but then we have other databases with genome assemblies like CottonGen, CGD, this is citrus genome database, and Cool Season Food Legume database. So a while ago, we came up with this system. So we use genus, species, genome, version, V, assembly version, dot, A, annotation version. So the example would be Prunus persica peach, genome V 2.0 dot point A1. So genus, species, obviously the genus species of the organism, and then the word genome, and then V is the assembly version. So we have like major and minor numbers. So it's like 1.0 if it's minor change, 1.1. And then if it's bigger change, we will go to 2.0. And then A is annotation version. And this is a single numeric value that is incremented each time a new annotation is released. But then it will start at 1 each time the assembly version is incremented. So even if the same annotation is used for version 1 and version 2 assembly, for the version 2, we start from A1, okay? So it works most of the time until we accumulated a lot of genome assemblies. So for example, right now, GDR has 21 genome assemblies from 7 crops and 14 species, and we cover like it's not like a single community like apple, peach, strawberry. They are all different, and so it is really difficult to coordinate. But then for example, like Prunus persica, Prunus avium, so it worked fine. But then like we found multiple genome assemblies from the same species, and in this case, like for apple, version 1 to 3 were using heterozygous golden delicious. Then the next version was using the double haploid golden delicious. So they used this name. The authors GDDH13 is a tree accession name, I think. So and then they came up with a new, they couldn't really call it version 4 because it's the different material. So now we will have to, like Maize community, we will have to add the accession name, right? And then, like for some cases, same multiple genome assemblies with the same accession. So like in cotton gen, Gossypium hirsutum, AD1, the same TM1 cultivar, like 3 different institutions generated the assembly. So we added the institution name. But then in GDR, we are in this situation, the same accession, same species, same accession, same institution. But then somehow they split up and they published 2 different genomes. And so the first one they called, and we agree, and Rosa chinensis genome version 1.0. And then another paper just came out like not too long after. So they both used old blush, and it was homozygous. But then they used old blush homozygous, so we just used whatever they used. Then in the same paper, they did Illumina genome, so and then they called the Illumina genome version 1, and then the most refined, much better refined homozygous genome as old blush homozygous genome version 2. So this is not really version 2 of the previous version 1. So for now we are just using exactly the name exactly published. So we have this situation as well. And then we found that sometimes authors skip versions when they publish. So we have like Fragaria vesca version 1.0, 1.1, 2.0, but then the next one, they published it as 4.0, and the reasoning was just in case someone publishes 3.0. So, we just took whatever is published. And then, so I guess that is better, but then sometimes authors publish duplicated names. So this is with the annotation versions. So
Look, I'm just going to say you're coming up on 5 minutes here.
okay, so this is like the last one, I think. So the Fragaria Vesca version 1.0 and 1.1, and they did the annotation, and the same annotation A1 was aligned to the new assembly version 2.0. So so we called it A1, but then someone else came up with new annotation for the version 1.0, so we call them 1.1 A2. And then the 2.0 people actually aligned both A1 and A2, so we originally called them in GDR as 2.0 A2, but then another people didn't know that, and then they published it as 2.0.A2. So since we have to keep the published name, we just changed it 2.0A2, the previous one we called, as just like we put a track name with the publication. So this is the problem we encountered. So in conclusion, we really need to refine the naming system. Obviously, we need to add the accession name and probably institution name. And then to enforce it, we really need to work with journals and communities. So that's my last slide.
Yeah, we're all having the same issues here. So, wow. Okay, so Marcella will tell us about Ensemble, which also deals with a large number of genomes.
Okay, can somebody give me the ball or something? I'm trying to share.
What you do is you, at the bottom of your screen, there's a green share button.
You cannot start screen share while the other participant is sharing.
Oh, okay, so you have to unshare it.
I just unshared it.
Yeah.
Okay, I see the Grameen, there we go, I can see your thing.
All right.
Perfect.
Why is not?
Wonderful, I see it, Marcella, good.
Okay, cool. Well, I'm going to tell you about a little experience that we have working with so far 56 different species, and these go all over. These include monocots, dicots, and some basal plants as well. Whoa. We basically reuse the gene IDs that were assigned originally by the individual genome projects who are responsible for annotating and submitting to the INSDC. The INSDC, for those that are not so familiar, include the GenBank in the US, ENA in the UK, the DBJ in Japan. We, saying Grameen, Plant Reactome, and Ensemble plants work with the INSDC and mainly with ENA because that's where Ensemble plants is located to resolve IDs and prioritize UniProt that are UniProt IDs because they are protein-based whenever applicable. We adopt a system that was agreed and adopted by the community early on, preferentially. We provide also mechanisms, type of ID converter, or sometimes simple tables for mapping back to IDs in previous version whenever possible. And we see that model organism databases and other resources work as well with GenBank and ENA to get a picture of how IDs are constructed. So just to give you a flavor of what we deal with, here are some examples of a few species, including some of the ones that we already heard about, where we have the assembly, the most recent assembly for the given species. Well, we tried at least to get the recent assembly, and the gene annotation. As you can see, not always coincides the assembly with the gene annotation. It doesn't always, it is on the same assembly. The gene annotation does not always correspond to the same assembly. We consult with the community and try to get what is the best set. You also can see different examples for naming conventions. For example, you heard from the For TAIR, the 80, the genus and species, and you have others that use more letters for that. You have some cases where you have appended the version of the assembly and so on, a suffix to signify also versions at this level, not transcripts, because these are all gene IDs. So in the future, what we see is that all of these assemblies are going to stabilize. We're going to have a same assembly on this column, but the gene annotations can always be improved. It's going to be updated based on new evidence. We also, in addition of the genes, the gene annotations currently include protein coding, non-coding RNAs, pseudogenes, but also we need to figure out, we need to keep in mind the regulatory features that we're going to need to annotate. And for the table for all 56 species is in the release notes. In every release we update this. The challenges that we see, like other species, is a lack of continuity in gene IDs and names. We also caution on including too much information in a name. For example, the version number, we advocate for that not to be the case. Propagating gene information between versions of the reference assembly, because many of the approaches that we see, different approaches, are based on the quality of the draft assembly and the complexity of the organism. We also see challenges associated with the initial submission and then later updates submitting to NCBI. Not all proteomes are fully represented or don't get updated in UniProt. And more recently, we are moving to the pangenome concept from a single reference to many references. And it's all of the above issues, but a little more complex. We learned from the first experience, perhaps, with what's Arabidopsis thaliana and Lyrata. And now we're dealing with some cases like maize. We have the 24 NAM parents. We have grape, where the suggestion is to include species and cultivar names, which is being talked about, and I'm going to show you a little bit more about adding prefix and suffix letter codes to IDs across the species, trying to keep the same orthologous gene models. And keeping in mind that some gene models are going to be species specific and are not going to be translatable. But as you will see, this is a little more challenging. This is a little more detail on that strategy. I don't know if I have all the time to go into it, but you can see that for species, this is a proposal. They talk about, for species, adding the first 3 letters of the genus, the first 2 letters for the species, and then for the cultivars, you can have, for example, 3 types of grape wine grapes: Cabernet, Pinot Noir, and Flame, and they are all going to be VV because they come from vinifera, but then you would add a suffix for specifying that this particular gene comes from the cap. Same concord in another species of Vitis labrusca. This has its own issues because this is the workflow would require to rename and transform the IDs. So that's one of the issues that have not been mentioned so far.
Marcella, you're about 5 minutes. You can go. Yeah.
Okay. Yeah, just a couple more. This is just to show you that we provide, external reference. So when you're mapping from one resource to another, so it's important to have standards to be able to convert assemblies and cross-reference. In the tools. So there are suggestions. Once the data is released from NSDC, we should all try to use the same ID. We suggest to prioritize representing up-to-date proteomes and from UniProt. Gene IDs independent of assembly version, genomic location, function, orthology, all of that, to keep them uniform, unique. Adopt standards from existing the best quality nomenclature systems. Examples: Newman, the Saccharomyces, Arabidopsis, that have been pretty adopted from the very beginning and preserved because this will allow also to have a very good curated literature by preserving the genomes. Standard tools to convert IDs across resources, assemblies, etc. And this was one idea that we have been talking with Ensemble Plans, which is a community collaborative platform, a type of data wiki that lets communities provide their own names. But again, we may want to standardize even that. And this was just a stat that Dan provided to me about how from closely related species, in this case rice, can be projected from one to another. But I'll leave it there.
Thank you, Marcela. Thanks. Next we have Pankaj from Plantiam. So Marcela, you can unshare your, oh, you guys are ahead of the game. Great.
Okay. Can you all hear me?
Yes, and we can see your slide.
Right, good. I think so far I've seen a lot of strategies, and some have worked very nicely over a period of a couple of decades. So my goal or my role in nomenclature business came in when I started doing my postdoc in Saccharomyces, and that's the time when the genome came out, and they had pretty consistent gene nomenclature, no matter what you call it. It needs to have a social security number so that everybody can track exactly what you're talking about. It's all about consistency. And then followed up later on when the rice genome came out. So I was the one who drafted the rice gene nomenclature, the OS IDs that you see, or LOC IDs that you see, that's was done by me, and later on drafted it for strawberry as well, for the version 1.1 that Suk was presenting, and then the eucalyptus. Rice one was published and everybody's using it in the community, and that was built on the framework of what Tanya presented about Arabidopsis and Saccharomyces, how to handle much of the versions and isoforms and allelic isoforms as well, alleles as well, because we are not even discussing about it. So on that front, I think we've heard about that there are reference genomes that can come from one single germplasm or ecotype or an accession, or we can have a non-reference genome that's like 1,001 genomes from Arabidopsis, or similar ones from grape, or 3,000 from rice, and going up on maize could be 40,000, or the 25 new genomes that are coming in, mainly for SNPs. But if they have sequenced it nicely to a good coverage, then you can actually find new and novel genes that are missing sometimes in the reference genomes, or they may have some other isoform annotations. So, and third one is the pan genomes that majority are people creating at the species level, but at some point you will start seeing at the genus level as well. So Plantiam in that respect is looking at not at the genome level, but since we deal mainly with functional and phenotype annotation, so we're wondering more about the total set of gene set represented for a species or an ecotype or a germplasm that you can think about it. So what we have been observing, and please feel free to provide us an input, what do you see when the gene sets are changed or gene annotation is changed, especially in terms of structure. Many times you would identify a new gene based on the new transcriptome or proteome data, and that includes coding and non-coding genes as well. You may identify a null allele. For example, the maize B73 may have a gene, but W202 may not have that particular gene, so it could be a null allele. Or later on, when they sequence any of these new 25 varieties, they might see a gene there, but that is missing in B73, so it's a null allele in B73. So what do you do with that? So similarly, for existing genes, you would often see changes in the versions. These are the most common ones. Start and stop coordinates may change because you have an altered UTR. You may have a different transcript isoform that is just at the same locus, but changing in a different isoform. So nomenclature, what we have seen so far, has not discussed about the isoform nomenclature. So you need to consider that. And then obviously, if it is a protein coding, then you will see a peptide sequence might change based on the splice isoform. You would also see mergers. How do you do mergers? And they were very nicely described in the rice gene nomenclature, and I think that works. And it was the same for Saccharomyces and Arabidopsis as well. You would see sometimes, not too often, but sometimes you would see splits happening between the genes. So which one carries the original gene ID or gene model ID, and which one gets a new one. So at least you would have one new gene added, minimum of it. And then you would see obsoletes and deletes happening between the versions as well. So how are you going to manage it? This one is a complicated figure, but focus on here. What Plantiam is trying to do is a superset of genes that is independent of where it has been mapped. So you need to create a pool of sum of all the genes that are discussed in your germplasm or accession or taxon as a catalog of genes. We call it as Genepedia or Gene Wikipedia, whatever you call it. That gene, depending on the accession, may map to a genome, and sometimes if they're Mendelian genes, then you don't have those kind of stuff. So you need to have that pool over here. And the locus can change depending on which genome they are mapped onto, or which version. And then obviously the allelic isoforms, null alleles, and other isoforms that you would see. So these are the 3-4 main things that we will be observing. We're trying to put together in Plantiam, because what we want to do is we need to have a consistency for the community, not only for their referencing purposes and writing manuscripts and analysis, but we also want them to be, these community also extends to the bioinformatics ones for semantic querying between various other annotations provided by hundreds of other online resources. The biggest problem would come, and as I can imagine, with all the version numbers showing up into your sufficiently less annotated genomes, is if you have version numbers in your IDs and genes, then how are you going to map and you do the NLP? It's a total chaos which people are going to create that I've seen in these presentations today. And then data updates, how are you going to update the data, and how are you going to connect back to the same gene ID that you would be looking at? Because not every gene has a gene name or a symbol or a human readable some kind of a name nomenclature.
Pankaj, you're coming up here on 5 minutes.
Sure. So idea is what we are trying to get is super gene set, which is at the species or genus level. Populate the super gene set by adding uniques to the pool if you find new things. And then for each version of the annotation, not assembly, map to the super pool and then auto IDs and exactly what you would do. That's what I think Arabidopsis was trying to do as well. Never ever create a version dependent IDs in your pool, and make sure you deposit, no matter, you can have your another version, but if you don't deposit it to the GenBank or INSDC, as Marcella was talking about, then it is a completely ignorant way of doing it in that sense, because they are well equipped to take care of the version numbers, and they will always map to the original things. That's what is to be happening. Not every genome that gets sequenced gets deposited, or the annotations don't get deposited either. So one last one is, if the users are performing structural annotations to revise existing models, continue using the same gene ID at the locus. Release, and don't release it immediately. Release it with your new version. It can be periodically, quarterly or half-yearly or annual annotation releases. Just do it that way rather than just creating +1, +2, +3. Everyone needs to use the same set of updated gene sets. So if you have a second or third party annotations, they need to go back to the super pool, map it back, and then provide. If they find new things, then you revise and release it as a new version assembly. We have dealt with that problem. The problem was created in rice with 2 different gene annotation sets, and it's still not resolved. And I see that problem happening in Fragaria and other genomes that are coming in, and they have not learned anything from that. So, and if a new gene is added or create a new ID, so if it is a pseudomolecule assembly, what we decided was all these gene IDs, Arabidopsis and others, I think Arabidopsis uses the 10th space, rice uses 100, but depending, as we are discovering more genes, you can use that last 100 to 1,000 space to put that additional gene in that series. If you don't have that pseudomolecule offer, just go by what is the next available in the scaffolds, if you have the scaffold level assemblies. But always release it with new annotation and do deposit it in the GenBank. That's what we are going to be suggesting. But what Plantiam is trying to do is have a collective set of the superset that everybody should try to use it. That's all from me.
Great, thanks, Pankaj. And I just asked Tanner like last night, but I don't know. Tanner, do you want to say anything about the wheat genome?
Sure, Lisa.
Okay, thank you.
Sure. So I'm listening to all of this, and I realize that when it comes to wheat, we are coming to the genome game, so to speak, a bit late. So Arabidopsis genome was sequenced in 2000, and compare this with the wheat genome, which was fully sequenced in 2018, 18 years later. Of course, we do have challenges in the field, but I think these are common challenges to all of us. So we have technical challenges like data standardization, naming conventions. We also have sometimes challenges with respect to geography or authority. Essentially, who will have a say in deciding how the gene models or assemblies will be named. We also have cultural challenges. For example, especially in our field, people coming from a genomic perspective versus people coming from a more genetic background. So let me just touch just a little bit in each category, and then I think my 5 minutes will be up very soon. Wheat, first of all, it has a huge genome. It has 17 gigabases distributed over 46 chromosomes. So that's a challenge in itself. It also has similar chromosomes with respect to other Tritacea species, such as barley. So that also was another challenge we have. How do we name a gene in wheat that is also available in barley or in, because of genome duplication in other chromosome in the same wheat genome? So that's the challenge we have. When you look at the assemblies, we already have many assemblies. And when we look at who's doing these assemblies, it shows the international character of wheat. For example, if you look at international wheat genome sequencing consortium, there are 68 countries under that consortium. Many people are leading this at different aspects. People from France, people from Germany, Australia, UK, of course, the United States. But this genome consortium only focuses on Chinese Spring, just as one accession. There are also Edgelops, they're Wildhammer, Drumit, and these are being sequenced by people based in California or take the lead. Wildhammer is actually in Israel. Drumit, of course, is also called pasta wheat. Italians are taking charge of this genome assembly. And pangenomes, now there is one pangenome group in Australia, another pangenome group now located in Canada. So these are all issues that speak to authority or geographical distribution. We don't have one place in wheat that can go through the gene models, or that can say you have to have gene models in one way, or genome assemblies, you have to name gene assemblies in this way. So it is very distributed. So we are trying to take care of this problem by starting to talk about it. And we have a Genome Nomenclature committee under the WIT initiative and the WIT information system expert working group. And we have a group of people from Japan, from Italy, from England, to actually from CIMMYT, to get together and talk about how we can actually provide guidelines to for the standardization of gene names or gene model names. But it is a challenge. It's an ongoing challenge. And as I said, the cultural challenge, perhaps Arabidopsis may not have it, but maize, I know, has the same problem. People coming from a genetic background, what is a gene? Right question. And people coming from genetic background. And when we ask what is a gene, people actually answer differently. So wheat is a species that people are working on for decades, not centuries. And we do have something called wheat gene catalog, and we have this actually it has a counterpart in maize. In maize, we used to call it classical gene set, and it is the same idea. There are some genes genetically identified or characterized genes, and how we can carry this between, take this rich and important resource and then make connections with the genomic data. So just to recap, so we have technical challenges, we have authority challenges, and we have cultural challenges. Thanks.
That's a phew. Sorry, Tanner. Is that we're just coming up on 11? Is that can people stay 5 more minutes? Were you done, Tanner?
Yeah, we can.
Okay.
No problem.
Okay. So I have this is very interesting to me. So I have 2 questions and 1 comment. The comment is that just in my field, maize, there is heated, passionate debate about how to name a gene like Ligulus 1 or Rad51, or just not the gene models, but who gets to name the gene. And those debates have been going on for a long time. We have a nomenclature committee. It's gotten very messy in the last 10 years. But I think for genome nomenclature, since nobody is yet passionate about their way is the best way, I think we really have an opportunity to make a consistent standard nomenclature for genome assemblies and annotation sets that we could make this really consistent going forward. So I want to ask, is there a desire to do this? Do we have a desire to make a uniform nomenclature? So everybody can unmute.
Yeah, I do too.
Yes, I agree.
Yeah, it's
Okay.
where.
Yeah, okay. And if so, can we?
So I'm going to dissent. This is Stephen. I think it's a lost cause. I think everybody who spoke basically presented a different solution to the problem.
Correct.
I'd love a unified solution. I don't think we'll get there. I think it's worth trying, but I think we also have to live with the fact that there will be a multitude of solutions.
Yeah, I think it's worth trying and finding areas where we can agree, because I doubt that we will all be able to come up with exactly the same nomenclature, but if we can come up with some consistent conventions so that when you are looking at a gene model from a different organism than you're used to, you can decipher it, or a different genome assembly identifier.
Well,
let me jump in just for a second. This is David.
Hi, David.
Assuming that we actually did come up with a unified nomenclature that everybody agreed with, that would require every assembly and gene on the planet to be renamed. That's a very unlikely event.
Well, I think we can't really go back and rename existing genomes. That would just be chaos.
Yes.
But if we could come up with a system for new genomes moving forward, I think then we'd have a hope of getting it adopted. But going back and renaming everything in the past is not going to happen.
So what that would mean in soybean is that we would have, for the first 5 reference genomes, we'd have one nomenclature system. And for all future ones, we'd have a different nomenclature system. That's also unlikely.
But I think that's okay.
Well, it's not unlikely, David, because it's what's happening. I guess I would propose sort of a twofold solution. One is a set of recommended practices that we know some people will follow and some people won't, and some people will have to follow. And part two is basically a way of tracking provenance and aliases.
Yeah, I think we can at least come up with principles like add this and don't add this, or not really concrete system.
Well, and I also think we could be able to come up with a nomenclature for a genome assembly, a genome annotation set. I think we could come up with that because, see, and again, David, to your point, genomes are just getting better and better, and we're sort of right at the beginning of this. I think that the genomes we have now, in 5 years, we won't be using those anymore. We'll be using better ones. So those old nomenclature systems will be, they'll fall by the wayside. But going forward, if we, like Jody said, it would be great to have at least the genome assemblies and annotation sets consistently named. I think we could do it.
I think another area where we might be able to come up with something that's fairly broad is naming pangenes.
Yeah, I agree, because that hasn't really been established yet to...
Yeah, exactly.
Is genome nomenclature.
Yeah,
just know how passionate people are.
This is Tanner Shan. So I will need to go soon because I have another meeting, but I think this is an issue that we need to think and discuss more. Perhaps we can just dedicate one meeting just to discuss.
I think we should have a working group on this topic with people that are interested and lay out the problems, which we just had a nice example of all the issues, and then have a working group. And it's going to take more than one session, I think. Would people be willing to? I would be willing to serve on a working group to this effect.
Yes.
This is Doreen. I think that a working group is important for this, because as we're starting to, it's just going to become really critical that we are able to share data across the species, the agronomically important species.
Yeah.
And while I agree there's a lot of cultural issues associated with ownership of existing names and transitioning over, we really are at a time where more genomes are coming out, and a recommendation, a set of best practices, a set of shared software or shared methodology to help with, for instance, those transformations, the renamings that we're talking, and remapping and QC of those. Those are things that we can all share.
Yeah.
And being able to work together on it will actually get us much further as a group than each of us having our own separate versions of those, especially when it's coming to these remapping issues.
Hi, this is Tanya. I also want to add that while we have a fairly large group on this call today, I think it's pretty clear that we need to involve additional people in the discussion, because it is not just a US effort, right, to sequence many of these organisms. And the people who are doing the actual sequencing have a very large stake in this discussion, and we need to involve them as much as we can. And if we don't have the appropriate contacts yet, then we should strive to reach out and include those people as well.
Yes, that's great. And we may even want to have an advisor from NCBI or something like that.
Hi, this is Sushma from OSU, and I'm partly here also for the GRAPE community. So GRAPE has a kind of a community members site at the IGGP that is very international in scope. And GRAPE, as Marcella has presented, has also gene nomenclature issues, and we also came up with a white paper last 2 years back. And so I can communicate to the IGGP group. I had already sent them email, but perhaps this time was in conflict and it was a short notice this time. But we will try to bring more Gray folks here next time.
So I guess the first thing we have to do is establish a working group with all these, like the Wheat International Consortium that, we need to establish a working group.
Let's invite Phytosome as well.
Exactly, yes.
With people.
Is there anybody that would like to take on this working group?
I can help. This is Sushma from OSU.
Hi, Sushma. Okay. Do you want to contact, send an email at requesting people join this working group?
We need to first perhaps generate an email list, and I would like to have input from everybody because I may not be knowing
sure.
people, right? Yeah, but I can coordinate that. I can start Excel and share with you and other folks, and we can perhaps add, and then I can do that.
That sounds good.
Just to throw this out there, for naming like genes and stuff, NCBI has their system. A lot of annotation tools will name genes as part of the annotation process with a gene ID. I'm not talking about functional names, but gene ID names. But maybe part of this proposal or whatever we're proposing to do is also work with annotation pipelines to auto-generate like names of the genome annotations and stuff like that. We do a little bit of this with GenSAS. We try to hopefully help the users make the downstream process easier for them. So just something I wanted to throw out there to consider.
I think that's an excellent point, Jody. That's why I was recommending that many annotation versions and assembly versions are being produced, but many of them are not being deposited in the GenBank or and that is the biggest bottleneck because they have the tools in place to take care of many of the problems that we are facing. So one of the recommendations probably would come out is that no matter what to the journals, that you have to submit your assembly and your annotation.
You know how we deal with this in Maize is we just don't, if you're not going to deposit it to GenBank, we're not going to, it's just not going to get exposure. It has to meet our criteria. So.
Well, then GenBank now anymore, you submit your genome and maybe you submit gene IDs or gene names with it. They now have a tendency to put everything through their annotation pipeline. And
they have changed their nomenclature for Maize to match our nomenclature.
Okay.
Have a document.
Okay, good.
They are flexible.
So the one thing that I've observed, and Brian White from NCBI actually told me he's the only plant person, or maybe there are only 3 or 4, but he leaves that in.
Right.
Is that if you don't submit the annotation, NCBI annotates on its own. And then those gene models are very different from what the community may have produced. And
see now this we don't have to solve all the problems. That's
have to initiate discussion, I guess.
Right. And NCBI did re-annotate Maize. So we just show those as different tracks on our browser. As long as they have the right names, we just show all the tracks.
So
This is Gerard Lazo. One of the things about doing these annotations is you also want to make sure that it makes good connections to ontologies. GenBank has been very poor in doing these sort of connections, whereas Uniprot has some nice tools where they've developed Uniprot to go annotations. So this might be some avenue to try to explore or make sure that these connections can be made.
Good point. Yeah.
Yeah. So in Grameen, we do have the Go annotations also on the genes. And other thing I wanted to add is that this discussion can be very useful for the species which are new and non-model and are being sequenced and being generated. So for example, here we have collaborators in OSU who are working on the Mint genome or the hops genome. And we initiated a little bit of the discussion about the gene nomenclature actually in Pathway January. And they are very excited that if there are guidelines and people, for example, working in model species have a longer exposure. So I think they can directly benefit and we can prevent some of the mess that can get along with the newer species.
Yeah. I think people are not passionately associated with genome names yet, like they are with gene names. So if we could nip this in the bud.
Yeah.
Lisa, this is Laurel. I just wanted to make one real quick comment. We only have heard from plant groups here.
I know. Yes.
How are people dealing with this in the animal databases?
Well,
Monica here, we deal with insect genomes, and a lot of our genomes are just nascent ones. They've just recently been sequenced, and the users don't have a whole lot of opinions about it or a whole lot of guidance on how to do things. So we'd be willing to follow a lead. One question that I came up with when listening to all this, especially to these genomes with a lot of kind of historical baggage to them, I haven't heard any requirement, except I think Ethy alluded to, why do we need a name for a genome? What requirements should the name have? It seems to be both an accession, which it shouldn't be, that should be a unique identifier. It seems to include metadata, which I also am very uncomfortable with. I think the main reason we have it is as a shorthand for the user, for the scientist. So it'd be good if there is a working group to actually discuss those requirements clearly for each of the data types, assembly versus annotation, versus gene name, just to see if we can find a compromise that hits all the requirements well.
That's true, and we also have to define everything, like a gene gene versus gene model is a big deal in maize, but it's not in other species, and we need to make some definitions.
Yeah, I think it would be good if we could get some, particularly a model animal organism.
Yes, like mouse.
They have a pretty mature system and could maybe say, well, here's what we're doing, and here's what works, and here's what doesn't.
C. elegans would be another good model.
Yeah. What little I know about the zebrafish, they use the human names wherever possible, and I'm curious if livestock does that as well, where there are correlations.
I remember back when I was in grad school, it was like we did C. elegans, Arabidopsis, and I'm from a bacterial background where genes are very homologous, and so they always had the same gene names consistently across organisms pretty much. Now, not so much, but I always thought the eukaryotes were screwed up because it was the same gene, but in this one it was called peewee, and this one it was called this. So I think they have made some effort to try to standardize their stuff. But yeah, talking to them would be good because they may have some perspective.
Jody, they do have, I'm very envious of the human, they have a Hugo, a gene, I mean, those are like ADHD1 or, they have ADHD1. And they are pushing it out to other species, other vertebrates. And I really wish we could do that. That is a huge effort. And I want to make that separate from this effort. This effort is just genome because, and we can make gene model names. We can talk about that, but I don't think that I
agree. We should stay away from gene names, but gene model names, we probably, if we could have an input on that, because that is usually done when you annotate.
That's right. Exactly.
Right.
Exactly. I'm also a little leery about, I think that different organisms might have different requirements for what is a homolog and an ortholog. And I think we could do it just independent of that.
Yeah, but if a gene ID should be unique, so it wouldn't affect gene IDs. It'd only affect gene names if there was orthologs and stuff like that.
Okay.
Yeah.
Whew, well, yes, definitely needs more. We need to figure out a group that can do this, and a very inclusive group. We need to define what the issues are and what we want to tackle in the group, and then we have to start working. So, and this is, I think this can be under the grant. We may or may not write this in the grant, but I think this is a really good example of what AgBioData can do, and this would be a huge feather in all of our caps if we could have this as a deliverable. I think this would be really fantastic. Are there any other comments? Phew, this is a big issue. Thank you so many people for joining, and We will talk next month. Next month we'll have Mike Cherry telling us about their efforts.